Understanding the role of SMAD4 in pancreatic cancer. Hello, this evidence-based content is prepared by Saboor fellowship-trained consultant surgeon, and if you or someone you care about has been diagnosed with pancreatic cancer and you've heard about genetic testing or biomarkers, you're in the right place. I know when you hear terms like metastasis patterns and genetic testing, it can feel overwhelming, but today we're going to cut through the complexity. By the end of this video, you'll understand exactly what the SMAD4 biomarker tells us about how pancreatic cancer behaves, why this specific test matters for treatment planning, and what questions to ask your oncology team. Before we dive into why this biomarker matters so much, let's quickly establish what we're dealing with. The SMAD4 gene, formerly known as DPC4, is located on chromosome 18q, and it's essentially a communication system inside your cells. Think of it as a messenger that carries signals from the cell surface to the nucleus, specifically from transforming growth factor beta receptors. When SMAD4 is working properly, it helps regulate cell growth and prevents abnormal cell division. But in approximately 50% of pancreatic cancers, this gene gets inactivated, meaning the brakes on cancer growth get released. This matters to you personally, because understanding your tumor's SMAD4 status could fundamentally change how your medical team approaches your treatment. Here's where it gets interesting, and why your doctors care so much about this particular biomarker. Pancreatic cancers with loss of SMAD4 expression have higher rates of distant metastases and a poorer prognosis. Research demonstrates that the SMAD4 gene status of the primary tumor correlates directly with patterns of failure in patients with pancreatic cancer. According to a 2024 study by Anstat and colleagues, SMAD4 loss predicts worse overall and distant metastases-free survival in patients with resected pancreatic adenocarcinoma. Let me break down what patterns of failure means. If your tumor has intact SMAD4 expression, it tends to recur locally near where the original tumor was. But if SMAD4 is lost or inactivated, the cancer is significantly more likely to spread to distant organs like the liver, lungs, or other parts of the body. You can see why this information is crucial. It fundamentally changes the surveillance strategy and treatment approach your team will recommend. If you're supporting someone through this diagnosis, understanding SMAD4 status helps you grasp why certain treatment decisions are being made. A tumor with SMAD4 loss might require more aggressive systemic therapy, because we're fighting cancer cells that have a higher propensity to travel. Now you might be wondering how this gene actually stops working. There are three main mechanisms. In 30% of tumors with SMAD4 inactivation, the gene is knocked out by homozygous deletion. That's when both copies of the gene are completely deleted from the DNA. In another 20% of cases, there's a somatic intragenic pathogenic variant. That's a mutation that develops during your lifetime, not one you were born with, coupled with loss of the second allele. Here's something that might surprise you. SMAD4 isn't the only player in this signaling pathway. Somatic mutations in genes coding for other components of the transforming growth factor beta signaling pathway, such as SMAD3, TGF-beta-R1, and TGF-beta-R2, have also been reported in pancreatic cancer, underscoring the importance of this entire communication system to cancer development. If this applies to you, and your tumor has SMAD4 loss, this is information to discuss with your oncologist about what it means for your specific treatment plan. But here's where this becomes actionable medicine. Immunolabeling for the SMAD4 protein has been developed, and the results of immunohistochemical labeling of tissue sections using this antibody strongly correlate with SMAD4 gene status. When pathologists examine your tumor biopsy under the microscope, they can apply special stains that reveal whether SMAD4 protein is present or absent in the cancer cells. This test serves multiple critical functions. First, immunohistochemistry for the presence or absence of SMAD4 protein can be used diagnostically as an adjunct to the interpretation of difficult tissue biopsies. Second, and this is fascinating, it can suggest the pancreas as a possible primary site in patients with metastatic adenocarcinoma of unknown primary site. Here's a real-world example. If a patient had a pancreatic cancer in which SMAD4 is genetically inactivated and later developed an adenocarcinoma in the lung that showed intact SMAD4 expression, it could be deduced that the tumor in the lung was a separate primary cancer, not a spread from the pancreas. This distinction fundamentally changes treatment decisions. If you're worried about someone you love going through multiple biopsies or confusing test results, SMAD4, immunohistochemistry, is one of those tools that can clarify the diagnosis when other tests leave questions unanswered. While further studies are needed, these data suggest that SMAD4 gene status may someday be useful for prognostic stratification and therapeutic decision-making. Currently, SMAD4 testing primarily informs prognosis. It helps your medical team understand how aggressive your particular tumor is likely to be. But we're moving toward a future where SMAD4 status might directly influence which chemotherapy regimens you receive. how intensively you're monitored after surgery, and whether you're a candidate for specific clinical trials. The transforming growth factor beta signaling pathway that SMAD4 is part of represents one of the major control systems in cancer development. Research into targeting this pathway therapeutically is ongoing, and your SMAD4 status might eventually determine eligibility for novel treatments specifically designed to address cancers with this particular molecular defect. If you're supporting a family member through pancreatic cancer treatment, understanding that SMAD4 testing is part of the broader movement toward precision medicine can provide context for why multiple genetic tests are being ordered. So here's your bottom line. Let's talk about the 3 categories of action you can take. Request comprehensive molecular testing. If you or someone you care about has been diagnosed with pancreatic cancer, ensure that SMAD4 testing via immunohistochemistry is included in the pathology workup. This should happen automatically at specialized cancer centers, but it's worth confirming. Call your pathology department and ask, was SMAD4 immunohistochemistry performed on my tumor? What was the result? Connect with a specialized center. If your initial diagnosis occurred at a community hospital, consider consultation at a high-volume pancreatic cancer center, where molecular testing and interpretation are routine. These centers typically have multidisciplinary tumor boards that specifically discuss how biomarker results should influence treatment plans. Come prepared with specific questions. What is my tumor's SMAD4 status, and how does this influence my treatment plan? Based on my SMAD4 status, am I at higher risk for distant metastases or local recurrence? Should my SMAD4 status change the frequency or type of surveillance imaging I receive after treatment? Are there clinical trials specifically designed for patients with SMAD4 intact or SMAD4 lost tumors? How does my SMAD4 status interact with other genetic findings in my tumor, like KRAS mutations or HRD gene alterations? If you're told your tumor has SMAD4 loss, ask about the implications for systemic therapy intensity. The data suggests these tumors behave more aggressively, with higher distant metastasis rates, which might justify more intensive chemotherapy approaches. Immediately, if your pathology report is unclear about SMAD4 status, or if SMAD4 testing wasn't performed. Not all pathology laboratories automatically include this in their pancreatic cancer workup, but it should be standard at comprehensive cancer centers. Within 2 weeks, if you're trying to decide between treatment options and SMAD4 status hasn't been factored into the discussion. This biomarker provides prognostic information that should influence therapeutic decision-making. For second opinions, if your oncologist dismisses the importance of SMAD4 testing or suggests it doesn't matter for your treatment plan, while the therapeutic implications are still evolving, the prognostic information is well established and should be part of your comprehensive assessment. Remember, SMAD4 testing is just one piece of a larger molecular puzzle. Your tumor should also be tested for KRAS mutations, TP53 alterations, and DNA repair gene defects like BRCA1/2 and PALB2, all of which have direct treatment implications. SMAD4 status provides complementary information about cancer behavior and prognosis. Let me quickly recap. SMAD4 loss in pancreatic cancer signals higher risk of widespread disease and changes how your oncology team should monitor for recurrence. Your next step, confirm your tumor's SMAD4 status through immunohistochemistry testing and discuss the implications with your healthcare provider. This information is for education. Always discuss your individual situation with your healthcare provider. I have outlined several reputable resources here which you may find valuable for further information on this topic. All links are available in the description below. Thank you for watching.